7 hours. You hit the target. Your tracker confirmed it. Your sleep score was good. But a controlled experiment found something your app is not showing you. Researchers disrupted only the deep sleep portion of a full night, leaving total hours intact. And Alzheimer's proteins in the cerebrospinal fluid were measurably higher the next morning. Total sleep time showed zero correlation with the result. Only one specific stage drove it, and it is not the stage your app tracks. In the next 10 minutes, you will know what that stage is, how to measure it, and exactly what to do about it tonight. That stage is called N3, slow-wave deep sleep. One of 4 distinct stages your brain cycles through each night, not REM, that most people are familiar with. Here is what a meta-analysis of 65 sleep studies across 3,577 people found about it across the lifespan. At age 25, approximately 20% of a full night is N3. By 50, that figure drops to around 12%. By 70, it falls below 10, often reaches 6 or 7. Not because those people sleep fewer hours, because the internal composition of their sleep changes. The architecture collapses inward, the hours stay the same. If you are past 35, this decline has already started in you. Not hypothetically, predictably, measurably, independent of every hour your tracker logs each morning. But here is the critical distinction between this biomarker and your age or your genetics. Sleep architecture is a quantifiable, adjustable target. The evidence supports specific interventions that move the needle. 3 of them cost nothing at all. By the end of this episode, you will know 3 things. First, what your personal N3 target should be, and the 3 methods for measuring whether you are hitting it right now, from a free wearable estimate to a clinical-grade home study. Second, why this specific stage is the operative variable and total sleep time is not, confirmed by the controlled experiment we just referenced, with the mechanism explained. And third, this one is counterintuitive. It requires you to stay for the protocol section. The nights you feel you are sleeping most deeply may be doing the most damage to your brain's waste clearance system. The reason involves something most sleep advice never addresses. We get there in the 3rd tier. Let's start with the biology. Once you understand what your brain is doing during N3 sleep, the evidence will make immediate sense. Your brain is metabolically expensive. Roughly 2% of your body weight, consuming 20% of your calories. That metabolic activity produces waste, proteins and byproducts that accumulate in the fluid between your brain cells throughout the day. Your body has a system for clearing them, and it runs primarily while you sleep. Animal studies, specifically mouse models from researchers at the University of Rochester, demonstrate that during sleep, the space between brain cells expands by approximately 60%, and a flow of cerebrospinal fluid moves through, clearing metabolic waste, including amyloid-beta, one of the proteins associated with Alzheimer's disease. This system, called the glymphatic system, is most active during slow-wave sleep. During N1 and N2, the process is minimal. During N3, it is fully engaged. Here is the finding that reframes the entire conversation about sleep duration. A study of 10 healthy adults, using hourly cerebrospinal fluid sampling across a 33-hour period, found that amyloid-beta levels in the CSF fluctuate predictably across the day and night. The peak, measured at 8 to 10 in the evening, was 27.6% higher than the morning draw. This was a small sample of 10 male participants, and these findings should be understood as directional. But the pattern is clear. Amyloid-beta rises during wakefulness. It clears during sleep. Every hour you spend awake when you should be in slow-wave sleep is an hour the system is not running. This is not a population correlation. We have direct experimental proof of the architecture-specific mechanism. In a controlled crossover experiment, researchers used acoustic tones to selectively disrupt slow-wave activity during sleep, while leaving total sleep time, REM sleep, and sleep efficiency intact. The participants slept their full hours. They simply did not receive the slow-wave depth those hours were supposed to contain. The next morning, their CSF amyloid-beta was measurably higher, and the effect was specific. Total sleep time showed no significant relationship with amyloid levels. Slow-wave disruption alone drove the result. This was a small study of 17 participants. The finding is important and mechanistically precise, but it awaits replication in larger samples, not how long you sleep, what your sleep is made of. And the evidence extends across years, not just overnight. A longitudinal study followed 32 adults over a mean of 3.7 years, collecting PET imaging scans of amyloid deposition at multiple time points alongside baseline sleep polysomnography. Those with lower slow-wave activity at baseline showed greater amyloid accumulation on subsequent PET scans, and independently, higher tau burden. Duration was not the predictor, slow-wave architecture was. Again, 32 participants. The longitudinal design is a strength, the sample size is a limitation. The relationship between N3 sleep and amyloid is not one-directional. It feeds back on itself, and a third study shows exactly how. In a study of 26 older adults, researchers measured amyloid burden in the medial prefrontal cortex by PET imaging. They recorded full-night polysomnography and administered an episodic memory task before and after sleep. Higher amyloid burden in the prefrontal cortex was associated with attenuated slow-wave activity, specifically in the memory-relevant 0.6 to 1 Hz frequency range, and that reduction in slow-wave quality independently mediated worse overnight hippocampal memory consolidation. Amyloid disrupts deep sleep. Disrupted deep sleep allows amyloid to accumulate further. 26 participants, a small study from a high-impact journal, and a finding that has since been extended by larger work. This is the feedback loop, and based on the normative data, it begins in your 30s, not your 60s. There is one more finding worth understanding, because it changes what poor deep sleep actually means mechanically. A study of 52 adults, 20 young and 32 older, found that age-related prefrontal atrophy produces something specific during NREM sleep. The slow oscillations, the large brainwaves that define deep sleep, still occur in older adults. Sleep spindles, the faster oscillations that coordinate memory transfer, still occur, but their timing relationship is disrupted. In young adults, spindles arrive at the slow oscillation peak, the precise moment the hippocampus and cortex are synchronized for memory transfer. In older adults, spindles arrive approximately 46 degrees early on the rising flank, a shift of milliseconds that misses the transfer window entirely. And that mistiming independently predicted worse next-day episodic memory in the older group. The spindles are still firing, the architecture on its surface looks intact, but the timing has shifted enough to break the system. Now that you understand what N3 is and why it matters, the question is, how do you measure yours? 3 options in ascending order of precision. Consumer wearables, rings, watches, bands, detect sleep stages through heart rate variability and movement. A controlled validation study comparing multiple consumer devices against laboratory polysomnography found that they perform well for detecting total sleep time and sleep-wake state. For identifying N3 stage specifically, accuracy is substantially lower. That study included 34 participants, a limited sample, but the directional finding is consistent with the broader literature. Use wearables for directional trend tracking. Are your architecture percentages improving over 4 weeks of intervention? Not as clinical staging tools. Your target, 15 to 20% N3 of total sleep time. Consistent readings below 10% are a signal worth investigating further. Home sleep tests, EEG-based sensors, available through direct order platforms, approach clinical staging accuracy at significantly lower cost than in-lab studies. $150 to $300 per test. This is the minimum credible source for a validated N3 baseline. Clinical polysomnography, in-lab, full-night PSG, remains the gold standard, increasingly available through sleep medicine clinics directly, with or without physician referral depending on your region. Now you know the track, it's time to look at the stack. Here is what to do with your number. The free tier. The strongest evidence base for improving slow-wave sleep is aerobic exercise, and it costs nothing. A meta-analytic review of 66 studies, where 23 out of 25 regular exercise studies were randomized controlled trials, found that moderate-intensity aerobic exercise consistently increased slow-wave sleep duration, with the largest effects in previously sedentary individuals. The protocol the data supports: 3 or more sessions per week at moderate intensity, zone 2 pace, conversational effort. If you watch the VO2 max or HRV episode, this is the same training zone. The longevity evidence converges from multiple directions. The second free intervention is thermoregulatory, and the mechanism is worth understanding because it is not what most people think. A warm shower or bath taken 1 to 2 hours before bedtime does not improve sleep by relaxing you. It works by physics. Warm water on peripheral skin triggers vasodilation. As blood flow to your hands and feet increases, heat is dissipated, and your core body temperature drops faster. That drop is one of the primary signals your preoptic hypothalamus uses to initiate and sustain slow-wave sleep. In a controlled experiment using a thermosuit to manipulate skin temperature during polysomnography-monitored sleep, a 0.4 degrees Celsius increase in peripheral skin temperature suppressed nocturnal wakefulness significantly and shifted sleep toward deeper stages. 24 participants, with the most pronounced effects in elderly and insomnia groups, and confirmation in younger adults for proximal skin warming. Bedroom temperature at or below 68 degrees Fahrenheit supports the same process throughout the night. Third, elimination. And this may seem counterintuitive to how you feel. A meta-analysis of 27 studies found a clear dose-response relationship between pre-sleep alcohol consumption and REM suppression, beginning at 2 standard drinks or fewer. Here is the nuance that most sleep advice never explains. At higher doses, alcohol actually accelerates your entry into N3 sleep in the first half of the night. Many people report sleeping deeply after drinking, and that effect is real. The N3 entry is faster, but REM suppression continues throughout the entire night as blood alcohol clears. The nights that feel like your deepest sleep may be your most architecturally damaged. You get faster N3 entry and sustained REM loss. The architecture disruption is not recovered. The free tier protocol. No alcohol within 3 hours of sleep onset. The $50 tier. The primary value here is monitoring. A consumer sleep tracker at the Oura Ring Generation 3 or equivalent class gives you nightly directional data on N3 and REM percentages. Use it as a trend tool, not a clinical instrument. The question it answers, are your free tier interventions moving your architecture over 4 weeks? That signal is meaningful even without clinical grade staging precision. For supplements, one double-blind RCT found that 500 milligrams elemental magnesium daily for 8 weeks significantly improved sleep quality scores, sleep onset latency, and sleep efficiency compared to placebo. This was a single trial of 44 elderly adults with primary insomnia. The magnesium form used was magnesium oxide. These findings cannot be generalized to middle-aged or healthy adults without further study. If you supplement, magnesium glycinate and citrate have higher bioavailability than the oxide form studied here, though sleep-specific RCT evidence for those forms is thinner. Dose and form selection should involve a healthcare provider. The $200 tier, home polysomnography or PSG study. $150 to $400 for a validated home sleep test gives you the credible N3 and REM baseline against which every intervention can be measured. If your wearable is showing consistent low N3 readings and you want to know whether that signal reflects your actual architecture, a home PSG answers that question with clinical grade staging. It is the only tool at this price point that can. Episode 6. Sleep Architecture. Retest timeframe, 28 days. Evidence rating, 3 out of 5 stars. Strong cohort and experimental evidence that N3 slow wave composition, not total duration, predicts neurological aging markers. Tier 1 meta-analytic support for all 3 free tier interventions. No large randomized trial has yet demonstrated that improving N3 percentage reduces amyloid burden in humans over time. That causal chain is not complete. Free tier. Bedroom at or below 68 degrees. No alcohol within 3 hours of sleep onset. Aerobic exercise 3 or more times per week at zone 2 intensity. $50 tier. Consumer sleep tracker for directional N3 trend monitoring. Magnesium supplementation. Single small RCT, elderly insomnia population, preliminary evidence only. $200 tier. Home polysomnography for a validated sleep architecture baseline. There is one variable we have not addressed, and it may be the most important one. Everything in this protocol assumes you are completing your sleep cycles. But for an estimated 80% of people with sleep-disordered breathing, those cycles are being interrupted, sometimes 20 to 30 times per hour, before adequate slow-wave time can accumulate. Architecture optimization cannot compensate for that level of fragmentation. Next week, the biomarker that measures it directly, and why it may matter more for brain aging than any other single sleep variable.